Welcome to our channel. Today we're diving into the world of science to explore some of the craziest experiments that have changed the way we understand the universe. From the smallest particles to the nature of reality itself, these groundbreaking endeavors have fundamentally shifted our perspectives. Buckle up because this is going to be a wild ride. Throughout history, science has always been driven by curiosity, a thirst to unravel the mysteries of the cosmos and our place within it. And what better way to do that than through experimentation, pushing the boundaries of knowledge through careful observation and daring leaps of imagination. Get ready to have your mind blown wide open. Our first stop takes us into the bizarre realm of quantum mechanics, a world where the very fabric of reality seems to shimmer and shift before our eyes. Here we encounter the enigmatic double-slit experiment, a deceptively simple setup that shattered the foundations of classical physics. Imagine this. A beam of light, a wave of pure energy, is shone towards a barrier with two narrow slits. Behind the barrier lies a screen, ready to capture the light that passes through. Now, common sense tells us we should see two bright bands on the screen, corresponding to the light passing through each slit, and indeed, that's exactly what happens when we perform the experiment with everyday objects like marbles or water waves. But light, it seemed, had other plans. When scientists first performed this experiment with light, they were met with an astonishing result, an interference pattern. Instead of two bright bands, the screen was adorned with alternating bright and dark fringes, a telltale sign of waves interfering with each other. Light, it seemed, was behaving as a wave, spreading out like ripples on a pond. So far, so good. But here's where things get really weird. When individual particles of light, called photons, were fired at the slits one by one, the same interference pattern emerged. This was perplexing to say the least. How could individual particles, acting seemingly alone, create a pattern that implied they were interfering with themselves? The answer, as quantum mechanics later revealed, lay in the very nature of reality itself. Particles, it turned out, don't exist in a fixed state until they are observed. From the realm of quantum strangeness, we journey now to the depths of human psychology, where a controversial experiment conducted in the basement of Stanford University in 1971 continues to spark debate. The Stanford Prison Experiment, as it came to be known, was a chilling exploration of the power dynamics between prisoners and guards. Conceived by psychologist Philip Zimbardo, the experiment aimed to investigate the psychological effects of perceived power within a simulated prison environment. 24 male college students were randomly assigned roles as either guards or prisoners. Their lives transformed overnight. The guards, clad in mirrored sunglasses and wielding wooden batons, were instructed to maintain order and discipline. The prisoners, stripped of their identities and issued numbered smocks, were subjected to a dehumanizing process. What began as an academic exercise quickly spiraled into a disturbing display of psychological manipulation and abuse. The guards, emboldened by their newfound authority, embraced their roles with unsettling zeal. The prisoners, stripped of their autonomy, began to exhibit signs of extreme stress, anxiety, and emotional breakdown. The experiment, originally slated to last 2 weeks, was abruptly halted after just 6 days due to the escalating levels of psychological distress. The Stanford Prison Experiment remains a controversial and hotly debated study. Its ethical implications and methodological rigor subject to ongoing scrutiny. Despite the controversy, it serves as a cautionary tale, urging us to confront the darker aspects of human nature and to remain vigilant against the insidious influence of unchecked power. Prepare for a journey into the heart of matter, where scientists have constructed a colossal machine of unprecedented power and precision, the Large Hadron Collider, or LHC. Buried deep beneath the French-Swiss border, this 27-kilometer ring of superconducting magnets and sophisticated detectors is a modern-day marvel of engineering. Imagine a racetrack for the smallest constituents of matter, protons and other subatomic particles, accelerated to mind-boggling speeds. These particles, guided by powerful magnets, race around the ring in opposite directions, gaining energy with each lap until they collide in a burst of energy. But the LHC is more than just a powerful particle accelerator. It's a window into the fundamental building blocks of our universe. By smashing these particles together at such high energies, scientists can study the debris from these collisions, piecing together the secrets of the subatomic world. One of the LHC's most celebrated achievements was the discovery of the Higgs boson in 2012, a particle that had been predicted by theoretical physicists decades earlier. The Higgs boson, often referred to as the God particle, plays a crucial role in our understanding of mass. The discovery of the Higgs boson was a triumph of both theoretical and experimental physics, confirming decades of theoretical work. The LHC is a testament to the power of human collaboration, a shining example of what we can achieve when nations come together in the pursuit of knowledge. Our journey takes us now from the subatomic realm to the very origins of life itself, to a time when Earth was a young and volatile planet. In 1952, two scientists at the University of Chicago, Stanley Miller and Harold Urey, set out to recreate these primordial conditions in the laboratory. Their experiment, now considered a landmark in the field of abiogenesis, demonstrated that organic molecules could be spontaneously generated from inorganic precursors. Imagine a sealed glass apparatus, a miniature model of Earth's primordial atmosphere. Inside, a mixture of methane, ammonia, water vapor, and hydrogen gas swirled, simulating the chemical makeup of our planet's early atmosphere. Electrodes sparked, mimicking the energy of lightning strikes, while a heating element kept the water boiling. For days the experiment ran, the gases circulating, the sparks flying, the water vapor condensing and dripping back into the primordial soup below. And then something remarkable happened. The water, once clear, began to turn a murky pink, then a deep red. When Miller and Urey analyzed the contents of their primordial soup, they found a treasure trove of organic molecules, including amino acids. This groundbreaking experiment demonstrated that the basic ingredients of life could arise from non-living matter, under conditions thought to have existed on early Earth. Our final destination takes us to the cutting edge of biotechnology, where a revolutionary tool known as CRISPR-Cas9 has emerged. CRISPR-Cas9 gene editing allows scientists to precisely target and modify DNA sequences. Imagine a pair of molecular scissors capable of snipping DNA strands with exquisite precision. This is the essence of CRISPR-Cas9, a system borrowed from the immune systems of bacteria, repurposed as a powerful tool for genetic engineering. CRISPR, short for clustered regularly, interspaced short palindromic repeats, acts as a kind of genetic GPS, guiding the Cas9 enzyme to a specific location in the DNA. Once there, Cas9 can cut the DNA, allowing scientists to disable genes, correct genetic defects, or even insert new genes into an organism's genome. The potential applications of CRISPR-Cas9 gene editing are vast and varied, spanning medicine, agriculture, and beyond. In medicine, CRISPR-Cas9 holds promise for treating genetic diseases such as cystic fibrosis, sickle cell anemia, and Huntington's disease. Beyond treating inherited diseases, CRISPR-Cas9 could also revolutionize cancer treatment by engineering immune cells to more effectively target and destroy cancer cells. The technology could also be used to develop new diagnostic tools, create new antibiotics, and even engineer mosquitoes to prevent the spread of diseases like malaria and Zika virus. In agriculture, CRISPR-Cas9 could be used to develop crops that are more resistant to pests, diseases, and harsh environmental conditions. CRISPR-Cas9 gene editing is a powerful tool with immense potential for good, but it also raises ethical concerns. Thanks for watching. If you enjoyed this video, don't forget to like, subscribe, and hit the bell icon for more fascinating science content. Let us know in the comments which experiment you found the most mind-blowing. See you next time. From the quantum realm to the origins of life and the frontiers of genetic engineering, we've journeyed through some of the most mind-bending and groundbreaking experiments in scientific history. These experiments have not only expanded our understanding of the universe, but have also ignited our imaginations. The future of science, like the universe itself, is brimming with possibilities.